Hi, everyone. I'm Carol Fleck, and on behalf of the ADDitude team, I'm pleased to welcome you to today's ADHD Experts presentation titled The Gut-Brain Axis and the Development of Autism. Leading today's presentation is Dr. James Morton. Dr. Morton is an independent consultant specializing in biostatistics and bioinformatics. He previously served as an investigator at the Eunice Kennedy Shriver National Institute of Child Health and Human Development, and as an associate research scientist and research fellow at the Flatiron Institute of the Simons Foundation. He is the CEO of Gutsy Analytics. The exact cause of autism remains unknown. However, scientists are learning more about the gut-brain connection and how imbalances in the gut microbiota can potentially lead to a range of disorders, mental health issues, and neurological conditions, including autism, depression, Alzheimer's, and Parkinson's disease. Today, we'll explain what the gut microbiome is, its role in the potential development of autism, what emerging research may tell us, and possible treatment approaches in the future. We'd like to begin today's webinar by asking this poll question to our live audience. Have you observed an association between you or your child's diet and problematic behavioral patterns? Please select your answers and comment in the text box under the video player to tell us more. For answers to common webinar questions about slides, transcripts, and certificates of attendance, click on the FAQ tab of your webinar screen. If you're listening in replay or podcast mode, visit ADDitudemag.com and search podcast 480 to access the webinar resources, or simply click on the episode description wherever you stream your podcasts. And if you support the work we're doing here at ADDitude to strengthen the ADHD community, we encourage you to visit ADDitudemag.com/subscribe and sign up for ADDitude Magazine. If you subscribe today, you'll get our just published 25th anniversary issue that examines how far we've come in understanding ADHD and the advances in research and treatment that we expect to see over the coming years. ADDitude Magazine is having a 2-for-1 sale, so subscribe today for yourself or to share with a teacher or a loved one who could benefit from greater ADHD understanding. Click the magazine tab on screen to learn more. Finally, the sponsor of this webinar is Microbiota. The Microbiota probiotic kit contains 2 probiotics to support focus, healthy gut function, and normal dopamine and serotonin regulation. Microbiota Focus contains 6 strains that support focus, while Microbiota Mood contains 6 different strains that support mood and emotional balance. Quality probiotics provide your gut with the healthy bacteria it needs to support mental health through the gut-brain axis. Click the link on screen or visit felixhealthscience.com to learn more. ADDitude thanks our sponsors for supporting our webinars. Sponsorship has no influence on speaker selection or webinar content. So without further ado, I'm so pleased to welcome Dr. James Morton. Thank you so much for joining us today and for leading this discussion.
And thank you for having me here. Just to confirm, can everyone hear me?
Yes.
Okay, great. Before I begin this presentation, I do want to make a few disclaimers. As mentioned earlier, I am the CEO of a microbiome AI company, and this was contingent on some of the work that I'll be presenting today. And I also want to note that I am not a clinician. I don't have an MD. Therefore, I cannot prescribe medical advice. I cannot prescribe nutritional advice. And on top of that, I want to note that microbiome-based therapies are in very early stage research. I don't recommend trying any of these therapies at home. This type of work is highly experimental and could be dangerous. So if you are thinking about something along these lines, first consult a doctor and let them for advice. Instead, I'm going to talk about the current state of biotechnology research and give you a glimpse at what type of technical obstacles are inhibiting therapy development And it's important to note that we're not dealing with perfect information and that there is much unknown in terms of the underlying research and the cause of autism. But I can give you a glimpse of what we can learn about the connection between the gut microbiome and the context of autism after resolving some of these technical obstacles. And furthermore, I'll provide another glimpse of what microbiome-based therapies could be in the next 5 to 10 years. So let's jump into this topic and see where we are right now in this day and age. Right now, the human genome costs about $600 to sequence, which is several orders of magnitude cheaper than where it was 20 years ago, where it cost $100,000 per human genome. This trend outpaced Moore's law, which has been the trend of making chips for computers, so illustrating how fast this industry is growing. And because of the rapid drop in sequencing cost, we have learned much about not only the human body, but also the gut microbiome. We've learned that you as a person are more than just your genes. You are also comprised of trillions of organisms, and these organisms match your human cells one-to-one, and their genetic content is 1,000 to 1. For every single human gene, there are in the order of 1,000 microbial genes. And over the last decade, there have been a number of studies that link the gut microbiome to a wide array of disorders such as rheumatoid arthritis, inflammatory bowel disease, fatty liver disease, and many other diseases that were not suspected to have a gut microbiome signature, but in fact do. And one of these disorders is are disorders such as Parkinson's, schizophrenia, and other psychological disorders, including autism. But the problem is that the connection between autism and the gut microbiome remains largely unknown. But there is some hope given the recent developments on microbiome-based therapies. Specifically, there was a FMT study. FMT stands for fecal matter transplant. The idea here is that you take fecal matter from a donor and you transplant it into an individual. You wipe out the participant's gut microbiota with antibiotics, and you repopulate their gut microbiome using the donor's microbiome. And in this experiment, they conducted a clinical trial involving 18 participants, and they noticed that there was an improvement across multiple metrics regarding behavior, regarding GI symptoms, regarding motor skills, where we see that most of these participants had severe autism, and that that up to 2 years after the treatment, we see a decrease in autism symptoms that is on borderline minimal. But that being said, this study is one of a kind, that in the field, there's much disagreement about how the gut microbiome is linked to autism. And as a result, there has not been a consensus image that has emerged. And part of the reason is that we're dealing with a very complex system. The human body is comprised of organisms from the microbiome, human cells. There's an immune system. There are viruses, and the interplay between all of these different components is not well understood. And one issue is that there is a lack of consensus on how to appropriately analyze the data that measure all of these different quantities. And so what we did is we came up with a new framework that to be able to reanalyze as much of the existing data sets there is using a consistent approach to see what we can learn from all the publicly available data. And so what we did is we took a 4-step approach. First is we took all the published autism data sets that have microbiome, all the microbiome data sets that in the context of autism, and we ran our algorithm to identify differences between autistic individuals and their neurotypical counterparts. We then correlated the microbiome against so-called cytokines, which are immune markers, to see how the gut microbiome is linked to host immunity and inflammation. We also linked the gut microbiome to metabolites, and then further cross-linked the metabolites to human brain pathways to see how the microbiome could be indirectly linked to brain activity. And then finally, we see how the microbiome is linked to diet. And then with all of these pieces together, we then tried to answer the question, given what we can learn from the publicly available data, how do our findings match the results from the FMT study that I just showed earlier? Altogether, we analyzed 25 data sets collected across 18 studies that encapsulates 600 ADHD children, measuring 6 different omics levels. When I mean by omics, I'm referring to data sets like the microbiome, representing the collection of microbes, the virome, representing the collection of viruses, diet, immunity, the host brain transcriptome, which measures the gene expression in the human brain, for instance. Now, analyzing all these data sets jointly does pose multiple challenges. The first one being is that these data sets are comprised of counts, that they ultimately have incomplete information, that we don't necessarily observe all the microbes in the stool sample or all the viruses in the stool sample, or we don't observe all the cytokines. Furthermore, absolute concentrations are not available, which does pose problems in our analytical capabilities. But I do want to point out that when clinicians conduct these clinical trials, there is substantial effort into conducting these studies, recruiting participants, and handling all the logistical challenges of collecting data. And so it is safe to say that these clinicians foresaw many of the problems regarding so-called biological confounding. They measured age information, they measured sex information, both of which are known to confound diagnosis and microbiome measurements, but the statistical capabilities to incorporate this information is largely lacking. And that was our major innovation, that we developed a novel algorithm that can address all of the problems highlighted here. I'm not going to go into all the details into what happens behind the scenes with these algorithms, but I will give you a glimpse of what kind of problems we're dealing with. When we are identifying which microbes are different between these two populations, we run what's called a differential abundance analysis. The idea being is that donors will give us their poop, their stool, and then we will extract some of the bugs from the stool, and then we'll perform sequencing on it. The problem here is that when we are doing the sequencing, we don't observe all of the microbes in the stool. We only take a small scoop of it. And because trying to measure all the microbes is prohibitively cost intensive. And so the question is that given a small subsample of the bugs that we observe, can we identify what bugs are different between the two populations? Answering this question can allow us to identify microbes that could be that could be causing symptoms and that explain the ADHD physiology that we observe. This problem, while it appears to be simple, is more complex than you think. The reason being is that when we take these measurements, this is akin to performing a voting poll before election day. We don't observe for instance, we don't observe the entire population of the U.S. Every time that we conduct a poll. We take a poll of, a few hundred to a few thousand individuals. And as a result, we have partial information. We don't know how many microbes there are for every gram of stool. And ultimately, this gives us an incomplete information about what could be happening, what microbes could be increased or decreased between the individuals that we are interested in. I'm not going to go into the underlying algorithms, but I will highlight that we have workarounds to resolve this ambiguity. But this is just one problem of many. When we collect samples and we sequence them, there's other sources of confounding, such as the type of technology that you use. Right now on the market, there's two types of sequencing methodologies. There's what's called amplicon sequencing that allows you to sequence just a small part of the microbial genome. This will allow you to observe more microbes because you're not sequencing the full genome, but at the same time, this does have its own biases in the sense that you might be missing certain bugs if you sequence the wrong part of the genome. There's another approach called metagenomic sequencing, where you can sequence the entire genome, so you get all of the genes that are present in the bugs. The problem with this approach is that it's typically more shallow. Because you're sequencing a lot more genetic information, you might not be able to observe more rare organisms that are so-called lower abundance. And because these two different technologies are so different, it can be difficult to compare the two. This is an example that you might see in scientific publications. Every dot here represents a stool sample, and the distance between the samples represents how dissimilar they are. The takeaway here is that all the samples in red are shotgun metagenomic samples, that they look very different from the samples in blue, the 16S samples, the amplicon sequencing samples. And because of this difference, these samples are not comparable, even if they're collected from the same stool samples. To make things more complicated, sequencing experiments also have batch effects, where if different labs sequence the same samples, you might have differing results. As highlighted here, we have two samples highlighted, sample 4, sample 6 on the left, highlighted in orange and blue. You can see that same thing. You see that they are separating, that there is a strong difference between them. But then you also see strong differences with regards to the different labs that are processing these samples. And as a result, it's difficult to identify consistencies between the labs, because if your results depend on which lab sequence your results, it's difficult to replicate the findings. I'm going to jump ahead to the final section on technical obstacles regarding differential abundance analysis. What I've discussed up to this point have been primarily focused on technical obstacles, but there's also a biological confounder. For instance, autism males are diagnosed at a 3 times higher rate compared to autism females. Furthermore, the gut microbiome is changing rapidly during early childhood development. We see that as children progress to solid foods all the way up to adolescence, their gut microbiota is also rapidly changing, largely reflecting the changes in the dietary habits. And so accounting for differences in age, differences in gender are key. And unfortunately, most of the statistical methodologies are not tuned to account for these factors. And as a result, when the current statistical methods analyze these data sets, they compute group averages. They take the average autistic individual, the average neurotypical individual, and they'll try to compare how their biomarkers, their gut microbiota, how they differ. And the problem with this approach is that we know that autism is a wide spectrum. It's a very heterogeneous condition, and every child's different. And so what we propose to do instead is to use what's called age-sex matching, where we matched individual kids to neurotypical kids that are similar in age and have the same sex. And by doing this approach, we can have more granular comparisons. Altogether, with these components, we designed what's called a Bayesian differential ranking algorithm. This allowed us to remove the biological confounding so that kids with similar age and sex are compared to. We also removed and ignored experimental biases that I showed earlier regard to the so-called compositional bias, or the lack of absolute abundances. We, as in addition to sequencing protocol bias and the batch effects that I discussed earlier. And ultimately, our algorithm provided uncertainty estimates, so we can not only tell you what microbes are different between the autistic individuals and the neurotypical controls, but we can tell you how uncertain we are. With all this technical machinery aside, this algorithm allowed us to generate very granular results in terms of what microbes are different between the autistic individuals and the neurotypical controls. So on the left, we have a plot that measures the so-called log-fold change between ASD individuals and their neurotypical controls. On the top, you have these bar plots that represent the width, the uncertainty of the log-fold changes, and then the y-axis represents the actual value of the log-fold changes per microbe. The x-axis represents all of the microbes. We have 2,000 microbes sorted on the x-axis, and you can visualize the raw data and see the differences yourself in this heat map, where we took ASD individuals, their age-sex matched neurotypical control, and when we subtracted their entire microbiome profile. And then we can see highlighted in red the microbes that are more present in autistic individuals compared to microbes on the left highlighted in blue that are more present in the neurotypical individuals. Furthermore, we can build classifiers that distinguish autistic individuals from neurotypical controls with very high accuracy. We can see that out of the 9 studies that performed age-sex matching, 6 of them had clinically relevant classification accuracy with a so-called AUC greater than 0.87. This shows that we can accurately distinguish between autistic individuals and neurotypical controls just using the measured gut microbiota. Now, furthermore, when we take a look at the microbes that were predicted to be associated with autism, and compared them with microbiota predicted to be associated with the neurotypical controls, we can see that they all separate, that there's a strong difference between the neurotypical controls and the autistic individuals, as highlighted in this plot. Here we visualize what's called a log ratio, where we take the bugs on highlighted in red on the plot on the left, and then we compute the average of those bugs, and we take the ratio of the bugs in the control, and then we compute those differences between ASD individuals and the neurotypical controls. And we can see that there is a strong difference between the autistic children and the controls across all the studies. And then we not only see differences in the gut microbiota, but we also see differences with regards to the immune response, the cytokines. And an example is shown here, where we have on the left, on the y-axis are the log-fold changes for each microbe. And then on the x-axis, we have the log-fold changes due to cytokine concentration. This cytokine here is called IL-6. This is linked to pro-inflammatory cytokine. This causes inflammation. Every dot here represents a microbe. If you focus, and then we can see. We can also look at how the microbes respond to another cytokine called TGF-beta, which is also linked to inflammation. Highlighted in blue is Prevotella capri. These bugs are known to be sensitive to what's called Western diet. So they are differentiated with regards to the carbohydrates consumed, and as well as refined sugars. And looking at the plot here, we can see that while these bugs are slightly preferential towards neurotypical controls, that they are highly associated with, negatively associated with inflammation, meaning that they can lower inflammation. These bugs are potentially healthy bugs to look for. We also see in orange, these are called bacteroides. These bugs are more positively associated with inflammation, and they are more prevalent in autistic individuals. And then finally, we have Bifidobacterium. These bugs are important for breaking down breast milk. And one of the hypotheses with autism is that that autistic individuals could have a delayed development regarding their gut microbiota, which is why you see Bifidobacteria more often. In this particular case, we see that it's dead in the middle. So it's hard to say what the role is, but we do see that they are also negatively associated with inflammation, which is also a promising sign that these bugs could also be promising probiotics to look for. Finally, I do want to note is that when we take these bugs and we correlate them directly with the concentrations of IL-6 and TGF-beta, you do see a strong correlation, highlighting that these bugs are indeed strongly associated with immune response. Finally, we did a comparison of the gut microbiota compared to human brain pathways. What we did here is that we mapped, what I mean by a pathway is that these are the mechanisms that break down products that we digest. And when I refer to microbial pathways, I'm referring to the pathways, the enzymes in the gut that break down dietary compounds. And when I'm referring to human pathways, I'm referring to enzymes responsible for the breakdown of products in the brain. And the surprising part here is that even though the brain and the microbiome are two very distinct entities, we do see a very strong overlap, meaning that there are up to 95 common pathways between those detected in the gut and those detected in the brain. And these are primarily broken down to 7 categories: amino acid metabolism, vitamins, carbohydrates, lipids, nucleotides, sulfur metabolism, and mineral absorption. And the part that was very exciting was that what we see in the microbe-brain connection, we also see in dietary trends. Here we did a co-occurrence analysis mapping microbes that are commonly present in the presence of certain dietary compounds. And so we do see that there is a strong connection between the gut microbiome and dietary intake, and that the trends that we saw earlier are also reflected in the dietary deficiencies of these ASD children. That many of the ASD children in one of these studies were deficient in key amino acids, key vitamins, and key minerals. And then finally, we cross-referenced our analysis to the FMT that I alluded to at the beginning of this talk. We saw that there was an improvement in behavioral symptoms, as well as an improvement in GI symptoms and motor skills. And the part that we were very excited about was that the microbes that we detected to be associated with autism were knocked out by the FMT up to 2 years after the FMT was administered, highlighting that there could be a link between the gut microbiome and autism. That if you manipulate your gut microbiome, maybe there's something to this. In summary, the associations between the microbiome and autism are real and strong. And the association between the gut microbiome and other omics levels regarding immunity, inflammation, diet, and brain metabolism indicate crosstalk, and that our findings mirror microbiome disruption after the fecal matter transplant. Those are our main findings from the paper, but causal mechanistic insights remain limited. To date, there are a limited number of FMTs conducted in autistic individuals. There needs to be more insights before this can be translated to the general population. With that in mind, I know many of you are asking, what can we do now with the current knowledge? There are 3 things that come to mind. First is that diet is important, and diet is more accessible to us. This is something that we can manipulate to achieve the desired outcomes. But I also want to caution against focusing primarily on behavioral symptoms. In terms of more tangible end goals, I argue that focusing on GI symptoms and sleep are 2 outcomes that are more accessible to the general audience, and that I argue that these are not only more accessible, but getting the right data is key. Keeping track of dietary logs, keeping track of bowel movements, keeping track of sleep patterns can help you as a parent figure out how to better manage your child's symptoms. And I argue these are all linked. If you solve your child's GI symptoms, maybe that can help your child sleep through the night. And I know that many of you are struggling on a regular basis with those types of problems. And I do want to point out that there is more papers coming out that are linking GI symptoms, sleep to autistic individuals, to autistic symptoms. That if your child has GI problems, has sleep problems today, the chance of your child also having behavioral symptoms is tomorrow is substantially increased. At least this is what this paper suggests. They developed an AI tool that shows that you can predict your child's behavioral symptoms with high accuracy, given just GI symptoms and sleep information. But that being said, I think that things are going to look very different 5 to 10 years from now, that 5 years is the typical length of a clinical trial, and that right now FMTs do show substantial promise, but there is considerable risk. There are people that have passed away from FMTs because we're still learning how to properly quality control fecal matter transplants. If you take a microbiome stool from a donor who has infection, that has severe autoimmune disorder, you could transplant those issues to your child, and I don't think that's the risk that many of us are willing to take. And it will take about 5 to 10 years of R&D before FMTs can reach the general population and obtain FDA approval. But I argue that we are approaching that this is in the horizon. In the meantime, watch out for ongoing clinical trials, and feel free to contact our email at hello.gutsyanalytics.com. We're putting together a mailing list that can help keep you up to date with these ongoing advances. And then finally, I want to thank you for listening, and I want to thank my team that put all of these analyses together. This was the result of a large international consortium spread across multiple countries and multiple institutions, and it was a pleasure with working with them. And I want to thank you for listening, and I'm open for any questions.
Hey, Dr. Morton. Sorry, yeah, Carol's gonna keep trying to join us, but for now, I will start going ahead and asking some of the questions that we've received from the live audience. Sorry to keep you waiting. So one of the first questions I will go ahead and ask you, just pulling up the list here, is, today we talked a lot about autism and the connection to the development of autism, but is there a gut-brain connection to ADHD? Have you seen that in the field of research?
Yeah, unfortunately, I haven't seen the data for it. I see this as an outstanding question in the field.
Mm-hmm. Interesting. And let's see.
Carly?
Yeah, we can hear you.
Great.
Okay, great.
Okay. I'm really sorry about the glitches. So
no problem. I only asked him just one question, which was about the gut-brain connection to ADHD.
Okay. As the parent of a child with autism, is there something I could have done differently to prevent this?
Unfortunately, we can't answer those types of questions yet. It's really hard to say. And I think maybe the only thing I would recommend is to don't beat yourself up on that.
Yeah, that's a common question we get, as you can imagine.
Yeah.
Another question, is the gut-brain axis something that can happen in utero or disorders developed after birth?
Well, there's definitely evidence of happening in utero. There's the recent publication linking aspartame to increase in autism risk. There's also the risk due to the key ingredient in Tylenol, and that is slipping my brain at the moment. Acetaminophen. Yeah, acetaminophen. These effect sizes aren't exactly large, but it's they do increase risk.
Are certain aspects of gut-brain health hereditary?
Well, there's definitely a hereditary component. We do know that there are about 10% of autistic cases that have a genetic component. There's disorders like Fragilus X, which we do know are caused by genetic mutation. But in terms of what role genetics plays in the microbiome and autism, it's not clear. It's a huge gray area. And in terms of genetic research, we're not very good at understanding so-called polygenetic disorders. It could be due to a number of many genes rather than just a single gene mutation.
Someone asked, which disruptions in the brain connection cause psychiatric pathology and how, and what other common conditions might be linked to this kind of disruption?
I'm very sorry. I missed the beginning of the sentence. Do you mind if you could repeat it one more time?
Of course. Which disruptions in the gut-brain axis cause psychiatric pathology and how?
Unfortunately, I don't have the answer to that question either.
Yeah.
Otherwise, I'd be we'd be going straight to clinical trials. But if we do figure that out, that'd be very promising. But there is hope with the FMTs that we are seeing improvement in behavioral symptoms in 17 out of the 18 children 2 years after the FMT. If we understand better how the FMTs work, that could be a viable route. But ask me again 5 to 10 years from now.
Okay, we'll do that. How does imbalance in the gut microbiome show up in the body?
Unfortunately, this audience is asking me a lot of really hard questions that I don't know. But I can try my best. So we can only provide glimpses of what could be happening. So for instance, what we do see a disruption in amino acid metabolism and diet and brain metabolism, as well as the gut microbiome. And this could be important because we know that amino acids are key in both maintaining the immune system as well as the construction of neurotransmitters that enable communication between the neurons. So amino acids are important. How exactly to modulate that? How exactly to make sure that you have the proper balance? I don't have the answer to that.
Okay. Can changes in diet help adults with autism and children, or only children?
Unfortunately, the literature is quite scarce, and there's conflicting views on it. And so far, I haven't seen a strong effect size with regard to dietary interventions. And I think part of the reason is because encouraging kids to have a dietary intervention is difficult, given that they're typically picky eaters and they're highly selective. So I don't have a great answer for that either.
Is there a connection between the gut microbiome and autoimmune diseases? And I think that's an interesting question because you talked about higher inflammation in children with autism. So does that translate into autoimmune diseases? Or what does that imply or potentially predict for the future in these children?
Well, I'll say that's the one thing we can say with stronger certainty, that there's definitely a link between gut microbiota and immune system. When that the immune system is keeping the gut microbiome in check and preventing the bugs in our gut from eating us alive inside out. It's also it keeps us in check to make sure that the gut microbes are breaking down the dietary compounds as needed. So yes, there's absolutely a connection between gut microbiome and autoimmune disorders, but how exactly the two tie into each other, that's not fully characterized given how complex both the immune system and the gut microbiome are.
Someone asked, how does maternal diet during pregnancy influence neurological development or predisposition to autism or ADHD for that matter?
I would say it's very important. Unfortunately, carrying out clinical trials on pregnant women is also very challenging. So we don't have great answers, but from what we do see with actinomycin, with aspartame, there's definitely and with obesity, right? So if you have type 2 diabetes during pregnancy, if you're obese, your risk of having autistic individuals also goes up. So yes, it is very important. But unfortunately, I don't have recommendations.
Okay. A couple parents write, what can we do to support gut health in our children with autism?
I'm looking for the same answer, the same question. We're in the same boat there.
Yeah. Okay. My daughter has autism. I'm certain that gut and metabolic issues play a part in her behaviors. What kind of specialist should I see?
I think the key word there is a GI specialist. They can give recommendations on how to better regulate your children's GI symptoms, bowel movements, and they could probably give better dietary recommendations than I can.
This question is from a therapist who says, how can we as therapists educate parents on the link between the gut and the brain in a manner they can understand? What recommendations can we offer parents to support their children?
Yeah, this one's a tough one too, because we're figuring out how to educate ourselves. Unfortunately, I don't have great feedback for that either. But maybe the only thing I would point out is these two diagrams here. Maybe even the previous slide is that I think behavior is a very tricky subject. We don't have a great way to quantify it, right? It's very subjective. But I think as parents, there are things that we can tackle. We can tackle. We can better tackle GI symptoms, try to get constipation and diarrhea and other abdominal pain under control. We can try to make our kids sleep better, and then we can try to change their diet. But because there are just so much unknown, I can't give concrete recommendation on what to do. But I think maybe it is fruitful to consider outcomes that are more tangible to parents. If your child can sleep better through the night, your kid is probably going to have better brain development.
Does exposure to poverty, crime, trauma, and other difficult situations affect the gut-brain connection?
I'm also not an expert in that, but from what I've seen, yes. If you have additional stresses in your life, that is going to cause issues. If you can't afford the if you can't control your diet, then that's probably going to lead to other issues as well. But I think in terms of disparities, that is a very open-ended topic in terms of precisely how that impacts gut health.
We have a question about transgender children. Does gendered behavior impact the gut biome? Is it genetic or a combination of these factors?
So we do see that the diagnosis rate for autistic males is 3 times higher than females. So there is definitely a gender discrepancy. There has been putative suggestions that that transgender might have an impact as well. Exactly what that link is not clear to me.
Okay. Yeah, it is interesting. Someone asked the fact that the pattern of males with autism being diagnosed 3 times as higher, does that make it more of a social issue?
As in, like, because we preferentially diagnose males compared to females? I actually really like that question. We're really bad about bias, especially in the medical community. If you see some of the studies that we published, that we reanalyzed in our paper, there's definitely more males analyzed than females. And I personally think that is wrong, that we should be including more female subjects in our studies, because because of this, we have a worse understanding of the female physiology, which is ultimately it's bad, right? It's bad for the female population. That leads to issues with dosing in terms of the drugs to give to women. It leads to a worse understanding, and as a result, many women are actually overdosed. They're on drugs. I'm not the right person to talk about this because that's not my expertise. But from what the evidence I've seen is definitely enough to get me concerned.
Would you say that microbial stool analysis is a reliable diagnostic tool for autism?
Not at this stage, no. We try very hard to fix diagnostics on the data sets that we collected. And while there is promise, there are still too many technical obstacles that prevent us from building a diagnostic.
Okay. Is there a role, or can you explain the role of the vagus nerve in gut-brain communications?
I can highlight there's a lot of interest in it, that potentially appetite is modulated through the vagus nerve. That's from what I understand. I can't say much more than that.
Mm-hmm. Can you tell us what we might expect in terms of gut-brain connection research 5 to 10 years from now? What are some of the things you expect to see and the treatments that might come from it?
Well, if things pan out, we might have therapies 5 to 10 years from now. I don't think we're that far away from microbiome-based therapies, but it does require vetting. It requires better understanding of so-called mechanism of action. I think there's a glimmer of hope in this field.
So are you talking about medication or something else?
Either or. So I think 5 to 10 years, my prediction 5 to 10 years from now, that FMTs are going to become as routine as blood transfusions.
Wow.
And this will be treated, or even bone marrow transplants. I think this will become more commonly used as they become more safe. And I think a subpopulation of autistic individuals could be eligible for that. Mm-hmm. This does require a pretty major change of mindset in terms of regulation. So we've yet to see.
And controls as well, because you said it could be very dangerous, very risky.
Absolutely. And so I think FMTs is promising. I think emerging work with so-called LBPs, live biological products, these could be promising as well, where they, rather than getting a whole transplant of a donor stool, they culture a handful of bugs, they isolate a handful of molecules to give to a patient. It's much easier to quality control 5 bugs compared to 100.
Mm-hmm. I know that fecal transplants have been used in people with ulcerative colitis and Crohn's disease and other digestive disorders and diseases, but their success rate is like 50%. It's not great, and of course insurance doesn't cover it, so it's very expensive for the most part. Someone asked, what do pediatricians and psychologists say when they review the data you produce?
I haven't approached, well, I'll say that the pediatricians that I've talked to get very excited, especially the ones that are already in this field, because this mirrors many of the observations that they've seen in their research.
Mm-hmm. And someone asked, how early in an individual's life can gut microbiota be tested?
Testing, I mean, that can be done whenever, as soon as an infant starts having poop, or even during pregnancy. There's nothing stopping you from testing. Though I do want to note that most of the microbiome kits out there are not FDA approved. They're not quality controlled. So your mileage may vary.
So it's not something you would recommend at this time?
I mean, let's just say that we struggle analyzing these results. I think I'm not sure how equipped the general population would be to interpret those data sets.
Okay. Well, unfortunately, that has to be our last question because we're just about out of time. So, Dr. Morton, thank you so much for joining us today and contributing your voice to our ADHD community. We appreciate that very much.
Thank you for having me here.
Of course. And thank you to today's listeners. We hope to see you again for our next free webinar on November 29th. We'll discuss parenting styles that work for neurodivergent children with Dr. Caroline Mendel from the Child Mind Institute. Make sure you don't miss future ADDitude webinars, articles, or research updates by signing up to receive our free email newsletters at attitudemag.com/newsletters. Thank you, everyone. Have a great day.